The overall goal of this experiment is to observe the effects of Pseudomonas syringae PV tomato on redox-regulated proteins in tomato plants. First, inoculate tomato plants with Pseudomonas syringae PV tomato to induce pathogen infection and protein response. Proceed to stain with diaminobenzidine to observe changes in reactive oxygen species. Then, perform cis-TMT labelling in order to examine redox-regulated cysteines in proteins. Results obtained can measure changes between treated and control samples.
Hi, I'm Chen from Department of Biology and Proteomics Facility here at the University of Florida. The advantage of our cis-TMT tagging over existing methods such as isotope-coded affinity tag, also known as ICAD, is the ability to multiplex when analyzing redox-responsive proteins on a large scale. While this demonstration will provide insights into tomato redox-regulated proteins in response to pathogen infection, the experiment can be applied to other systems such as Drosophila, mice, humans, and other plants under control or stressed conditions.
We first had the idea for this method after we observed marked variation between ICAD experiments because only 2 tags are available.
Visual demonstration of this method is critical as the labeling and enrichment steps are difficult to learn. Experimental variations between labeled samples must be minimized and reducing reagents removed to ensure efficient labeling.
Let's get started.
Grow 2 transplanted tomato seedlings of similar size in 4-inch diameter pots. For the inoculum, culture a lawn of Pseudomonas syringae on a KBM plate. Scrape the bacteria into 20 ml of water and prepare a bacteria inoculum of around 10 to the 6 colony-forming units per milliliter in 1 litre of inoculation buffer. Dip the 4-week-old plants into the bacterial inoculation solution for 30 seconds. Then, cover immediately with clear plastic bags. After the preferred inoculation time, harvest the fully expanded leaf. Place epidermal side up in 1 mg per milliliter DAB stain and vacuum infiltrate for 15 minutes. Then, incubate leaves in the dark at room temperature overnight. Now boil the stained leaves in 95% ethanol for 10 minutes and store in 75% ethanol. Using a pestle and mortar, grind the harvested leaves to a fine powder in liquid nitrogen. Then, perform phenol extractions as described in the accompanying text. Next, precipitate phenol-extracted proteins with 5 volumes of cold 0.1 molar ammonium acetate in 100% methanol. Harvest the proteins by centrifugation. Perform pellet washes. Then resuspend samples in 1.5 ml of cold 70% ethanol and transfer to a 2 ml microcentrifuge tube. After centrifugation, discard the ethanol supernatant and dry the pellet briefly in a speed vac. Dissolve samples in a denaturing protein extraction buffer. After centrifuging to form a pellet, collect the supernatant and measure the protein concentration according to the manufacturer's manual. For each of the 6 mass tags, start with 100 micrograms of protein sample. Add equal volume of freshly prepared 200 millimolar iodoacetamide alkylation buffer to block the free thiol groups. Incubate at 37 degrees Celsius for an hour. Then, to precipitate the proteins, add 1 milliliter of cold 80% acetone and place at -20 degrees Celsius overnight. Pellet the protein by centrifugation and perform 3 washes with 80% acetone. After air drying samples on ice, resuspend the pellet in 50 microliters of lysis buffer. Then, to reduce the disulfide bonds, add 0.5 microliters of 100 millimolar TCEP and incubate for 1 hour at room temperature. Solubilize the tags by adding 20 microliters of acetonitrile to the reagent tubes. Vortex and spin down. Next, to remove extra TCEP, load 50 microliters of sample onto a Microcon 3KD column. Add 50 microliters of lysis buffer and centrifuge for 15 minutes at 10,000 times G and 4 degrees Celsius. To collect the sample, remove the column, invert into a clean tube and centrifuge. Check the pH of the sample. If needed, adjust to between pH 7 and 8 with 1 molar hydrochloric acid. Add 5 microliters of the cis-TMT reagent and incubate the reaction for 2 hours at room temperature. Combine samples and add an equal volume of Lamley sample buffer. Boil for 5 minutes and separate on a precast 12% polyacrylamide gel. After rinsing the gel 3 times with filtered water, stain with Kumasi blue for an hour and destain overnight in water. Based on protein band concentrations, divide 12 fractions per gel lane. Chop each fraction into 1 mm pieces and place into a 1.5 ml tube. Completely destain the gel pieces with 0.1 molar ammonium bicarbonate in 50% acetonitrile. Now cover the pieces with 100% acetonitrile for 15 minutes, remove the liquid and dry the pieces using a speed vac. Next, add an equal volume of trypsin solution and set on ice to rehydrate. Then incubate at 37 degrees Celsius for 12 to 16 hours for tryptic digestion. Next, remove the liquid protein extract from the incubated samples. Stop the reaction and remove any remaining protein digest by covering the gel pieces with 5% formic acid, 50% acetonitrile. Then, shake the gel pieces for 20 minutes at room temperature. Transfer the supernatant to the extracted protein sample tubes. After 2 more extractions, dry the samples under a speed vac. In 0.5 ml tubes, prepare 50% slurry of anti-TMT resin in TBS at a concentration proportional to the intensity of the respective protein band. Wash the resin 3 times with 1 column volume of 1 times TBS. Proceed to add 200 microliters of TBS to each sample. Then transfer to anti-TMT resin and agitate at room temperature for 2 hours, followed by rocking overnight at 4 degrees Celsius. Load each sample on a column. Perform column washes of 200 microliters each. Then, elute each sample 3 times with 200 microliters of 50% elution buffer and dry under vacuum. Resuspend the samples in 12 microliters of 3% acetonitrile with 0.1% formic acid and inject 5 microliters directly onto an HPLC column. Elute peptides using a gradient of increasing acetonitrile. As detailed in the accompanying text, use mass spectrometry to detect peptides. In a top 2x3 workflow, start with a single-stage MS, then acquire 3 MS-MS spectra with higher energy C-trap dissociation fragmentation, followed by 3 MS-MS with collision-induced dissociation for protein identification. Analyze the mass spec data using a branched workflow. Implement a reporter ion quantizer to quantify the ratio. Also, process the MS-MS spectra through the spectrum selector, spectrum normalizer, and spectrum grouper nodes. Query data against the sequest search engine. This image shows examples of a control tomato plant leaf on the left and a Pseudomonas inoculated leaf on the right. After the leaves are stained using DAB, the destaining process allows for the histochemical stain to show signs of reactive oxygen species in the leaf tissue. As expected, the control shows no significant staining, while the leaves treated with Pseudomonas stains positive for hydrogen peroxide production. This example shows proteome discover data output of a differentially redox-regulated protein. Peak area for each sample allows for absolute quantification. Peak area between control and inoculated samples indicates relative amounts. Here, redox regulation of a protein between a control and inoculated sample is similar. Peaks of similar intensity suggest the presence of disulfide bonds not regulated by a change in treatment.
Once mastered, this technique can be performed properly in 1 week. Remember to keep samples cold and keep labels organized. Also, use precaution when using phenol, such as nitrile gloves, and working in the hood.
Following this procedure, other methods like specific mass spectrometry analysis of post-translational modifications can be performed in order to determine other cysteine modifications like sulfonic acid and gluten cyanylation.
After watching this video, you should have a good understanding of how to perform DAB staining, cysteine labeling, and redox cysteine quantification.